Hi, hello everyone. Welcome to the single cell and spatial exploration group. So I'm Aaron from Bowel Turing. So today I'm very happy to share with you about the ability of the talk to data database in terms of screening and also in terms of identify the potential target for several diseases, including for today, I will focus on inflammatory bowel disease or IBD. And because in the talk to data database, we have several type of data, including single cell, bulk RNA, and recently we also have the public spatial transcriptomic database. So from that, we take advantage from these databases to help with the target identification, and then we can further explore more about biological function of the target to understand more about the target. And at the end, we can able to go to a public spatial transcriptomic data using spatial X for further exploring the context of that target in a pathological sample, for example. So from that, so I would like to go quickly to the agenda for today. So for today, the agenda. So first, we will include three main parts, and then followed by the summary and the discussion and the resource at the end. So the first one is I would like to show you this several approaches to narrow down the potential target. For example, you can get the target from the published database, and then you would like to use the data database that we have to able to narrow down the potential target. So we can do that as well. So from that, we can use like multi-omic database. So we can do with single cell, bulk RNA, and spatial data as well. And the second step is well, for the talk data, beside doing screening the expression of a gene, for example, through our different database, we also have tool and the AI to able to do different analysis to get different function from the target. For example, you can use AI Sonia to get more information about the target very quickly. And from that, you can able to deep dive into research paper that's involved in the target. And then we can do different analysis for some like co-expression, differential gene expression, and so on. And we also have recently we have the ASNAP function for you to explore a specific data set of your choice in talk to data. And then after that, we can further explore using spatial X to study the potential target as well. I will go quickly to this the spatial X section because usually we have the another section for spatial X. So the first one is go down to potential target. So for everyone to be on the same page, so I just quickly overview about the disease that we focus today. So which is the inflammatory bowel disease. So IBD is the inflammatory bowel disease, or IBD is referred to a group of like chronic disorder in which they marked by the ongoing inflammatory of the gastrointestinal tract lining, and the patient usually experience about abdominal pain, diarrhea, and rectal bleeding, and so on. So it is a very one of the popular disease. So and the two primary type of IBD include like Crohn's disease and ulcerative colitis. And over the survey of CDC in the US, we have around 2.4 to 3.1 million people have IBD, which is a big number. So to identify the potential drug target is crucial for advancing the progress for therapeutic development of IBD. So for that, we can go to the next one, which is usually to identify the potential target there's several ways, but the way that people usually find is useful and easy to use is easy to do is go to we could go to private or public database. So and recently people are quite exciting about the open targets database because this database is very comprehensive and include different resources which aggregate multiple published data resources into one place. And from that, they can give the scientists the way to prioritize for some target that could be potentially become the drug target to eliminate any destruction from other targets as well. So for example, here is the list of the gene that I get for IBD from open target. Even though they open target already give a very nice gene list with the association score, and but we're quite still a lot of gene to explore, which is a very nice option because we have more option to explore the target. But for one of the things that you can do using the TOC2 data database is you already have the list of target for IBD, and then we can able to look into the data what of these targets look like in the disease and in the cell type that you want. For example, here you can for example, here I can get the top like 50 gene from open target of the gene that associated with IBD. And then I would like to do a quick screening on the TOC2 data single cell database to see the expression level of these gene in different conditions. Possible, I would like to investigate into different cell type. For example, here I have macrophage, T cell, B cell, and intestinal epithelial cell. And from this cell type, I choose like 3 different conditions. So we have normal, we have chronic disease, and we have ulcerative colitis here. So we can compare the expression level between the 3 conditions on each cell type selected. So from that, we in the TOC2 data, we can able to do that. So you can able to filter for the study that you want. So you can kind of screening a little bit of study because you don't want to include like all of like unrelevant study to your screening, and then you can do the filtering for anatomy, also cell type, and also for condition to build the custom tree metadata like this. And then you can able to enter all of these genes in a single page, and then you can able to look at the overview of the expression level of the top 50 genes from the open target list. And from that, you can see, you can identify some significant spot here. For example, you can see there one gene called BLCG2, which is, you can see is very high expression in ulcerative colitis compared to control. Other targets is not look that significant, but instead only this one gene that for the top like 50 gene list that show very significant increase in the expression level, and also show the expression increase in the Crohn's disease as well. So it makes it kind of like interesting. Maybe this target have some potential information or potential ability to become a target for the exploration. So from that, that is the single cell data, and then we also have the bulk RNA and the spatial data as well. So for that, my next question is, if I look at that gene in different database, where not I can like pre-produce the same result. So first I look at the bulk RNA, and you can see here this gene BLCG2 is actually also highly expressed in colitis and the Crohn's disease compared to the normal. And also interestingly, in the spatial data, which is recently put it on the TOC2 data, and you can see they also show the high expression level from the colitis compared to the normal, which is go through the 3 database from single cell, bulk RNA, and spatial transcriptomic database. We all observe the same indication showing that BLCG2 is highly expressed in the colitis compared to the normal control. So from that, I can it looked like this target is very interesting. So I would like to understand more about what is the function of BLCG2 and any gene that relate to BLCG2 as well. So for that, we can use different tools in TOC2 data to be able to do the thing. So for example, I first would like to understand a little bit about the basic of that gene. you may have very well known about knowledge about the gene, but in case of you not very familiar with the gene, so what thing we can do is we can switch to the AI Sonia option in the talk to data, and the first option here is to give you the answer, like the large language model to answer you. And and I would like to mention this AI Sonia is specifically trained for biomedical research, so it's more focused on biomedical research to give more accurate answer on that. So for example, I would like to summarize the no function of this gene in intestinal epithelial cell, because you remember the one that actually show the upregulation is actually only in intestinal epithelial cell, but not in immune cell or macrophage. So it's very one of the interesting fact here. So that's why I focus on intestinal epithelial cell. And then and then we give you some information about the gene, including some like basic what is the gene look like. Is this an enzyme? And then they can give you some information about how BLCG2 actually involved in epithelial cell to inflammatory signal and pathogen. And from that basic information, so I can able to go further to find some research paper that involve in more specific information about how BLCG2 actually regulate some function in intestinal epithelial cell to involve in the IBD. So you can see one of the paper here that show intestinal epithelial cell is a barrier, is important for maintaining the mucosal immune homeostasis, and the dysregulation of this barrier will trigger the inflammation in the IBD. So which is a very nice connection between the cell and the gene and the function, that the disease that we study. Okay, so after I have some basic information using the AI Sonia, I can go next to explore further about maybe the next question that I would like to ask is any gene that co-express with that gene, and whether or not we know the function of this gene, whether or not this gene is actually involved in IBD or none of them is involved in IBD at all, which is we can kind of give indication whether or not we should focus on this target or maybe we should forget about that. So for example here, I look into the BLCG2 again, and you can see in this ulcerative colitis, and to identify the co-expression gene in the top data is very simple. We just simply click into that dot, and then the system will calculate the top 10 gene that co-express with BLCG2 using Pearson correlation. And then you can see the top 10 gene here. And we can, when I look at some of these genes, I actually 𝐾𝐈 is actually have some indication that involved in the IBD. For example, is upgraded of 𝐾𝐈 is activity in intestinal inflammation, and specifically in the IBD model. And also the MALAT1, which is a very famous non-coding RNA, is involved in many diseases and also include in the IBD as well. Okay, so after I have the explore about co-expression with the BLCG2, the another question that I would like to ask is whether or not we have where of this gene is expressed, even though we already have that expressed in intestinal epithelial cell from the gene query, but I would like to see more on the broad image of where it's expressed in the human. So for example here, what I can do is I can make atlas on top to data. So I can filter for insectin, and can filter for, ulcerative colitis. And then from that, I can create an atlas that contains the study with this filter. And within the atlas, I can see the cell type here, and I can query the gene that of my interest. For some is BLCG2. And you can see BLCG2 is confirmed actually highly expressed in epithelial cell in colon, which is a very nice way to look at the specifically of the expression of the gene on the cell. And this is you can do with the normal control as well, because that is one of the way we can identify some off-target cell that maybe if you develop a drug that target to BLCT2, which is the predicted cell population that could be affected from that inhibitor, for example. And the next thing that I can do is, beside the explore other gene that correspond with the BLCT2, and also what is the cell highly expressed, the BCLT2, I can also obtain the pathological slide with potential target expression pattern and relevant condition. So because taking advantage of the spatial transcriptomic data recently we have on the doctor data, so you can query the gene, and then you can see the expression of gene across different spatial slides here. And you can actually identify some of the slide that in the sample of your interest, and then you can open this slide or sample using SpatialX, which is the platform that specialize in analyzing spatial transcript. Platform, and then we can able to see how it's work. So okay, so I hope internet is okay. Okay, so this is the list of the target that I got from Open Target. So and then I can copy this list is IBD-associated gene. So for example, I copy the first 50 gene here, and then I can go to the talk to data. So this is the new user interface of talk to data, and then I can filter for the condition that I want. But for example, is this is I can go to single cell first, and there's several ways for you to able to filter. So for example, here we have the option to save the study that I interested, because I usually look at the study that more related into IBD, for example, and then can able to save that. For example, here I have the study of normal ulcerative Crohn's disease study, and then after that I just load that study into my filter, and actually you can able to adjust that by adding or removing the study using the study filter here as well. Okay, and after that I can go to the anatomy, which is intestine, and then cell type. I'm interested in T cell, interested in B cell, macrophage, and intestinal epithelial cell. And condition I'm interested in normal for sure, and Crohn's disease. And that one is ulcerative colitis. Okay, so now I filter everything that I want. And then the nice thing here is we have the custom tree view. So for the first level, I would like to choose the anatomy, which is intestine. The second level, I would like to choose cell type. And the third level, I would like to choose condition. After you update, you can see it's very nicely separated into cell type with 3 different conditions. And after that, to query the gene, I just simply copy from here and just paste into the gene query window here. And then you can see immediately it will give you kind of the real-time gene expression information immediately. And you can able to see across different 50 gene here, what is the expression level and what is the coverage of the expression. And you can able to spot out something that significant impact on this expression on these 3 conditions. And the one that I showed you before is BLCT2 here is actually very high expressed in ulcerative colitis, but low expressed in normal condition. And to do the co-expression that I just showed you before, what you can do is just simply click to this one, and they will calculate the top 10 genes that are highly correlate with BLCT2. So from that, you can able to download the gene list of your interest here, or get the heat map here. Okay. So that is the one very easy way for you to explore the data set on the top to data. And another one that I just showed you before is the atlas function. So in the atlas function, what you can do is we can able to filter for the study of your interest. For example, here I would like to filter for the ulcerative colitis. Do some testing first. And I would like to filter for one condition that I want. Okay, let me do one more time. Right. Okay. And then you can go to the atlas function here, and you can select the intestine and then create atlas. And then you can say that this is UC atlas, for example, and create. Yeah, it will take some time for the platform to collect all of the combine all of the study to able to make the atlas for you. And when you filter for this, actually you can able to go to the study to actually find the study of your interest here. You can just select the study you want. You think that is the best study that you want to include your atlas. You can able to select it here, and then you can able to customize that for your atlas. Okay. So one is running, so it's here as well. So you can see it will create the atlas with all the study that you created and give you UMAP here. And then you can see this we have all of the metadata, cell type, condition, and so on. You can able to explore that. For example, I just choose a few cell type for this purpose because it's gonna be loading time. So I give some cell type here. Okay. Okay, and then for example, you can intersect in a gene. You can able to do so as well. And you can query gene. And you can see the cell that highly express BCL2 is actually in the column epithelial cell. And then you can able to query different like multiple gene. You can see it's a gene list here. You can query multiple gene and you can able to see the expression pattern of these gene in different cell type. Okay, so that is the one that you can do with the analysis tool on top to data. And another option here is the AI that I mentioned before. So they have different mode. Here you can have the ask mode to ask the basic question that I just asked before. What is to summarize the function of the gene that I'm interest. We have agent mode. I will not cover today, but in the next section we already covered that. So agent mode is able to run the analysis to give you the result, just like you do the code expression using the user interface. You can able to do here as well by using natural language. And we have the other option to do search study and to do search gene through different databases. For today, I will cover the one that I just mentioned before. So we can just simply enter the information here, which is a very, I think for me, it's very convenient tool. So you have to switch between different platforms. You just go here and then you search, and then you can able to explore multiple steps using this information. And then you can see when you click into the gene, you're able to access to the data in the database as well. Okay. All right, so let's go back to the flow that we have here. So after we identify some gene and some relevant biological function of the potential target that we focus, which is BLCT2, so we can further explore about that. So for you can using the LM to ask more information about the gene. For example, we can see that BLCT2 highly expression will activate BKC and also 𝐾𝐈 activation, and that's good into the pro-inflammatory cytokine, for example, IL-6 or TGF-alpha, for example. It could amplify the immune epithelial crosstalk in IBD. So that could be one hypothesis that we can use AI to able to generate, and we can able further confirm with a scientific research paper using the information. And the second thing is The second thing is the second hypothesis is BLCT2 can hydrolyze BIF2 and to produce like IB3 and that. So BIF2 increase the calcium release from ER, while that can activate the BKC. And while with the high calcium can also modulate the tight junction turnover and other like actin, cytokeratin, and vehicle trafficking as well. So for example, the excess amount of calcium and PKC activation might disrupt the tight junction, which is leading to the increased permeability, which is a common observation in IBD. So this is could be a very nice way to generate some idea, and then you can verify that with scientific paper and research on the available publicly. And one thing that we can able also do in using our database and also the data in the database here is you can able to further explore the impact of the BLCT2 operation in tight junction marker. For example, here we have TJP1 in Crohn's disease. So you can able to open like single data set, the published data set. For sure, is if you have some in-house data set to able to test for hypothesis, that is one of the best way that we can do. But in terms of if you don't have the in-house data set, you still can find some value information on a published data set as well. So you can able to use the spatial database for the doctor data. So you can, for example, here we have the normal and the Crohn's disease, and then I can able to first, very basic step first is query gene to see the gene expression level of BLCT2 in normal compared to Crohn's disease. And also I want to see the expression level of one of the tight junction marker, which is TJP1 in the two conditions. And it seems to be we can further able to quantify the expression level of the two marker on these two sample into two conditions as well, which is you can use that using SpaceX. And for that, I would like to show you the way to explore the SpaceX information here, the spatial data. So in the database, we have like spatial data here, and we have several ways to view the spatial data. So spatial data can view at the cell level, the single cell level, or the spot level, depending on the technology that we have. So Visum, we have spot, and for example, like Xenium, we can have like cell level. So with the single cell, you can just do the same at the single cell database, which is you can do differential gene expression. You can query gene BLCT2 as well. And then you can able to query the gene across the database, and then you can able to source for the tissue or the cell type that highly express BLCT2, for example. And the second option that you can have here is you can display at the image. So with that image, you can able to look at the expression of this LCT2 using different spatial slide. So let me do one more time. So beside just the average expression, you can able to see in terms of the you have the spatial information of how to view the data here. Okay, little loading here. So it basically will show you the result that I showed you previously. So you can see different. We have different Visum slide here. Then you can see which sample that have the gene that highly expressed and involved in the disease of your interest. For example, here I have the with this slide we have highly expressed of BLCT2 and in the Crohn's disease condition, which is maybe interesting to look at this study because we can find some value information on the published data set that we can further explore using SpaceX. So from that, so for some here, you can open a data set using SpaceX, and you can see on the slide, it's been combined and lined up here, and you can query gene, you can further do the expression level from this gene. So for that, I would like to go to the last part here, which is the summary of the today club. So for today, we are able to do a quick overview of how the TOC2 data can able to help you with identify potential target from a list of target gene that you can obtain from the published database or published gene set. And then you can able to do a screening using like multi-omic database on the TOC2 data. And then you can study some biological function of the BLCT2 and other gene as well. In the context of regulating a certain cell type, because for single cell, we can take advantage of the cell type information, and then using different tools that we have like AI, Sonia analysis tool, and also custom atlas in TOC2 data. And we then further explore the possible mechanism of how the gene that can regulate IBD using the single published spatial transcriptomic dataset using SpaceX, which is that I don't have like much time for today, but we have the follow-up section to explore that using the published spatial data. Okay, so this is the resource. So even though you're not about twin user, you can able to explore public data set on the TOC2 data as well. We have the human cell lab and also Tahoe 100 million for you to explore using this link. And for the resource, so all of the SCSE Club recording will be posted on about twin YouTube channel, and you can find on the information about the TOC2 data documentation and tutorial video as well shown here. So I think that is pretty much that I cover for today SCSE Club. So if you have any questions, so please feel free to enter to the chat box, and if any like time limit, I cannot answer all of these questions. So please drop your email. I can follow up with you for all of these questions. So from that, thank you very much everyone for joining the section today. So I will go to the Q&A to see if I'm able to answer some of the questions. One question is, can we perform cell-cell communication from spatial transcriptomic data and TOC2 data? So I think for a single data set, yes, we can, when you open with SpaceX, you can do cell-cell communication, which is we have the spatial cell-cell communication on SpaceX. So you can do that using like single data set. But for the cross database, it's kind of challenging because not all the data that have like cell type information. So from that, I think it's more about like depend on data set that you would like to look at. And thank you very much for that question. So combine multiple single cell databases. It is possible to combine multiple single cell databases. So I think so I rephrase. So I think the multiple studies for sure, like the one that I show using the custom atlas, multiple databases. So I'm not pretty sure that what you mean. Is this mean that you have your own data and you would like to combine with your published data, or you mean we have like several published data and you're going to combine together? So in terms of if you have your own data and you would like to combine with published data, so one thing that there's several things way to do. One thing is you can able to you can go to a request to the data license to download the data, and then you can enter that using a vProcess Pro. And if it's not answer you, so you can please reach out to us so we can have a further discussion of what is the best that we can do to support you. If I have the data set of my interest, I can create my own atlas. How long does it take to generate this atlas? So I think in terms of if that data set is available on TOC2 data, yes, you can do immediately. You just go and show the data and then create the atlas. And how long does it take to generate? I think it's not that long, because the one that I do is around 1.3 million cells, and usually it take like maybe less than 2 minutes to create. Yeah. So how does the tool handle integration across multiple spatial datasets from different study? Um, Integration across multiple spatial datasets. So I think I think I will need further clarification on this question. So maybe I can follow up with you about that, and when I can answer you, this really depend on what you're looking for. So yeah, I will follow up with that question. The one thing is my talk data does not have show spatial transcriptomic slide. Yes, so we will release that soon to everyone. So we still in the QC testing. So it's just a little bit like early announced, but we will release to everyone. So how many data sets is available? So I think for the single cell, we have more than you can go directly to the top data to see that information. So for single cell, you can able to see here more than 3,000 study, 200 million cell, and for the spatial, we have around more than 3,000 slide. Yep. Okay. What is 𝐂𝐈 𝐒𝐳𝐬 doing here in the spatial data? So I think 𝐂𝐈 𝐒𝐳𝐬 is in the plan that they can use 𝐂𝐈 𝐒𝐳𝐬 to able to analyze all the data set in the talk to data database, and even though even they can do the cross database analysis as well. So we have first in spatial single cell. Sorry, first we have in the single cell, but I will hope we will soon have the 𝐂𝐈 able to analyze other database and also across database as well, and we'll let you know. How many bulk RNA dataset is available? So the bulk RNA is we have like more than 651 study and more than 32,000 sample. Yes. What are the cell marker used for cell annotation in integrated study the atlas created? I think that is the depend really because in the talk to data database we kind of standardize on the cell type using the same cell name is the same cell type. So from that when we the cell type from the single database will based on the author cell type, and then when we combine based on the standardized data we can combine them together. Can you do an 𝐂𝐈 in upcoming section? Yes, we will have the AI focus on the next club. So yeah, we'll wait for that. We're looking for that. Talk to data does not have spatial transfer. Yes, so we will have that soon. So we'll let you know soon. Yep. Spatial transcript limit number of genes that we can analyze. How can we outcome it? So you mean that the number of genes that we can do analysis at one time? So okay, so let this question. So I need to talk with the talk to data team, and also I would like to get some insight from you. So please, can you please reach out to us, or we can reach out to you as well, so we can understand better, and then we can able to discuss with the team for that. So what method is used to calculate the coverage in the analysis? Are those fixed for all? So okay, so I think for this I also will talk with the talk to the team to see what is the default threshold that they apply for the coverage, and I will follow up with you as well. Okay, I think I try to finish all of the questions in the Q&A box. So thank you very much everyone for joining the section today. So yeah, thank you much for your time, and hope it will be useful for your research, for your work. and looking forward to seeing you in the next single cell and spatial exploration group. Thank you very much. Bye.